Hi, I'm Avi Perna. I'm application scientist for BioLegend. Today I'll be talking about choosing the right buffers for flow cytometry. Choosing the right buffer set for your flow cytometry experiments will help you generate accurate and reproducible results. And while this conversation is aimed toward flow cytometry and cell sorting, these principles also apply to multiomics and histology approaches. When you're selecting buffers, you're generally choosing from common preparative buffers and more specialized buffers that are aimed for accessing your antigens of interest. Selecting the right buffer for your experiment is guided by the nature of your scientific question and of your biologic samples. If you're working with a tissue like blood, bone marrow, or splenocytes that contains red blood cells, you may want to use a preparative buffer to remove them. LymphoPure is a density gradient that allows you to isolate PBMCs from a whole blood sample. That'll remove your red blood cells, platelets, and neutrophils. RBC lysis buffer will lyse your red blood cells from whole blood, leaving behind live leukocytes. And finally, RBC lysis/fixation buffer is a buffer that will lyse your red blood cells and fix the leukocytes. This is a preparative step for downstream intracellular staining. Surface staining is a simple procedure of staining your cells, then washing away excess reagent with cell staining buffer. Intracellular staining requires that you access intracellular compartments. Some chemical probes can penetrate the cell and reach those, but when staining with antibodies that are labeled by fluorochromes or oligonucleotides, you need to select the right buffer to access the right compartment, whether that be cytoplasmic or nuclear. Fixation of cells preserves their biologic state at that moment. It strengthens them for permeabilization and intracellular staining, and it supports efficient scheduling of the workflow in your lab. Common fixation agents are neutral buffered formalin, methanol, and ethanol. BioLegend CytoFast is designed for cytoplasmic staining, typically for cytokines. True Nuclear allows you to access the nucleus of the cell to look at nuclear transcription factors. And True Phos Buffer allows you to study phosphoepitopes in intracellular signaling networks. Cell analysis is often the final step for your complex experiments, so choosing the right buffer is key. While homebrew buffers can be effective, BioLegend buffers are manufactured under a stringent quality management system and quality tested for your specific application. You likely can use BioLegend buffers with other suppliers' reagents. However, BioLegend buffers are tested and validated using BioLegend antibodies and proteins. So when you combine them, you can be confident in your hard-won results. If you have any further questions about selecting the right buffer, please reach out to us by email or telephone.